Welcome to this next in a series of Stanford Inventor webinars. I'm really pleased to introduce our two speakers for today. Their topic is going to be gene signature to predict outcome on metastasis in liver cancer. And with me are Professor Dean Felsher and Dr. Renu Dhanasekaran. Let me introduce them individually. Dean is actually a professor in the Division of Oncology, Department of Medicine and Pathology here at Stanford School of Medicine. He's also the Director of Translational Research and Applied Medicine, and he's Director of Research for the Division of Oncology. Renu, and also Dean, he got his MD PhD in molecular biology at UCLA, and a BS before that at University of Chicago. Renu is a board-certified gastroenterologist. She's a physician scientist working on translational research here at Stanford, focused on liver cancer, liver transplant, hepatology, and gastroenterology. She currently works in Dean Felsher's lab, and so Dean and Renu will be sharing the presentation today.
So Renu today is going to talk to you about a finding I think will be of great excitement to anybody who's looking for ways to cure the disease liver cancer. Generally, my laboratory has been studying for almost 20 years ways to cure cancer. We've developed strategies that allow us to uncover novel ways to target cancer, to diagnose cancer, as well as to make predictions about specific aspects of the biology of cancer. Renu's a brilliant scientist and doctor who will describe to you in detail some findings that we think will be a great excitement to people who are particularly interested in coming up with new treatments and prognosticating liver cancer. Renu.
Thank you, Scott. Thank you, Dean. Hi, everyone. So I'm Renu, as they introduced, and I'll be today talking to you about a gene signature test to predict outcome and metastasis in liver cancer. Okay, so we'll continue. So I'll broadly like to talk about our mission here. Our goal, if you see any patient who gets diagnosed with liver cancer or any cancer for that matter, they all get boxed into one group as being one particular set of patients. But we know that based on their gene expression profile, there are multiple different subtypes within any particular cancer. And if we can exploit this gene expression profile to classify patients into different groups, then we should be able to get patients the correct treatment. And this is our overall mission. So in this presentation, I'll talk about first the problem, what liver cancer is. I'll introduce you to the team of scientists and physicians. We'll go over our solution for the problem and how our solution is better. I'll introduce some technical details on how we derived this solution, and finally end with pointing out some practical applications and potential for commercialization. So let's start with liver cancer. Liver cancer is currently a global crisis. It is the second most common cause of cancer-related deaths in the world today. Around 750,000 cases of liver cancer are being diagnosed every year, especially in sub-Saharan Africa and in Asia. In the United States, around 40,000 new cases are diagnosed every year, but it is important to remember that there's a 43% increase in liver cancer incidence in the United States. This has been reported in the last decade. We are exploring this. But compared to every other cancer that is actually decreasing in incidence, liver cancer is rapidly increasing. So currently, what do we do? How do we predict prognosis? We use tumor size as a predictor of prognosis. For example, if a patient has a tumor of 5 centimeters, they're eligible for transplant. But if it is, say, 5.5 centimeters, they are not. It might seem crude to you, but this is what we are doing. And tumor recurrence, or the tumor coming back, is quite common despite using these restrictive criteria. For example, if 5 patients undergo resection, as in, part of the tumor is removed, it comes back in 2 of them. And let's say we do a transplantation, that is, remove the entire liver and give the patient a new liver. Even then, the tumor comes back in 1 out of 5 patients. So clearly, this is a problem we haven't solved. So why do we need a test? For these reasons. 1, we need to do pre-op risk stratification. We need to decide who can safely undergo surgery and not have the tumor come back. We need to optimize organ allocation. You might be aware, but organ shortage is a big problem. There are thousands of people currently on the waiting list just waiting for an organ to become available, and we need to make sure this precious resource goes to the right person. We have to, of course, determine prognosis. We need to be able to look the patient in the eye and tell them what their expected life expectancy is. And most importantly, we want to develop novel therapeutics. So let me introduce our team at this juncture. We are a group of physicians and scientists. We're trying to solve this problem. Dean, as has been introduced to you, has been working for over 20 years. And I myself, I'm a board certified gastroenterologist. I have a clinic just for patients with early liver cancer. We've also recruited Dr. Brendan Visser, who's a surgeon, Dr. Kothari, radiologist, and Dr. Gentles, who's a bioinformatician and assistant professor at Stanford, to work together as a team to come up with a solution. So now let me go on and introduce our product. Our product is a prognostic gene signature. We have identified a short 19 gene signature which can predict prognosis in patients who have liver cancer. How we developed this is also very novel and interesting because we used a proprietary mouse model of liver cancer and compared the genes to liver cancer genes from humans to find a short signature, which can be then taken to the bedside. We are currently validating this and almost done with the study for prospective clinical trial for validation. How this will work. So when a patient gets diagnosed with liver cancer, we can do a biopsy safely, get a small piece of tissue, extract the mRNA from that tissue, run it through a next generation sequencing based 19 gene signature test, and stratify patients into either low risk or high risk. How does this help? I see patients like this in my clinic all the time. These are the questions they ask. Can I get a liver transplant? Do I need more frequent imaging? Or is this tumor aggressive and should I increase the treatment? And for all these kinds of questions right now, other than tumor size or number, I don't have great answers. And hopefully this test will help us to stratify these patients so we can answer these very important questions. What about commercialization? Is there any such test currently for liver cancer? No, there is no such test for liver cancer right now. But for other cancers like breast cancer, there is a test called Oncotype or Prosigna. For colon cancer, there's a Coloprint. And for prostate cancer too, there is an Oncotype. These are gene signature expression-based, biopsy-based tests. They cost around $2,000 to $5,000 per test. And if we were just to conservatively extrapolate that there are 750,000 cases and we go for $2,000 a test, even then it's more than $1 billion market worldwide. I'll show you a little bit of technical details on how we approach and how we derived at this gene signature. So we have two unique proprietary liver cancer models that Dean has developed in our lab. One of them develops metastatic cancer that spreads and one doesn't. So this gives us a great tool to be able to compare these two directly and find out which are the genes driving metastasis or aggressive cancer. So we did exactly that, and we found around 600 genes here. Next, we took those genes and compared it to around 1,000 genes that are significant in human liver cancer. We got this from around 650 patients from multiple studies. So when we crossed these two, we found that 19 genes were common between these two groups. So that's very significant because there seems to be a conserved genetic program, and it is a short list, so it should be easily translatable to the bedside. And I'll show you the power of the signature in a few studies here. You can look at two different studies that we are showing the survival curves diverge, and the patients who have high expression of our gene signature do really poorly, statistically very significant. And the box plot there is showing to you that the gene expression of our 19 gene signature is very high in patients who have metastasis. This shows the power of the signature. We went ahead and externally validated in a completely different cohort of patients from the TCGA data set of around 373 patients. And again, our signature performs fantastic. Our p-value is great. And if you look at the bar graph, it will show that our signature is able to predict tumor stage and presence of metastasis. So at this point, I've shown you that we've developed it from 650 patients and validated another 350. So that already gives us over 1,000 patient cohort. What we're currently doing is a prospective clinical study, which is the gold standard. We'll have to validate this going prospective. So this is what we are doing at Stanford. These are the steps in which we are doing this. We designed a study where we collect tissue and blood from all patients who are undergoing treatment for liver cancer at Stanford. We have a biorepository. We extract RNA and sequence it based on our gene signature. Then we correlate this with their clinical outcome to see if our hypothesis holds good. And it's ongoing study, almost complete though. We have collected 120 patients already, and by early next year, we should be done with the rest. And we already have very promising data. Here, I'm just showing to you that most of our genes are significantly overexpressed in the cancer tissue compared to the normal tissue. This is exactly what we expected, and we are able to validate this prospectively. And even more excitingly, we see that our signature is expressed much higher in tumors that have poor prognosis or bad aggressive features like vascular invasion and higher tumor grade. So I'm confident that once we have the clinical outcome data, this is going to prove to be very beneficial in a prospective study too. Briefly, I'll just introduce that we are also doing other functional studies where we had generated patient-derived xenografts in mice where we can grow a library of different human tumors in mice, and then we can test different kinds of drugs for genes we identified in our gene signature. So we have ongoing functional human xenograph-based studies too. So currently, I would like to conclude by saying we can predict the prognosis of liver cancer, we can identify new therapeutics based on our gene signature, and this 19 gene signature will hopefully make it to the bedside, and we're looking for commercial partners who will enable us to do this.
So we really appreciate that you spent the time looking at this webinar and presentation. Renu and I are delighted if you contact us directly. OTL is glad to connect us, and information will be provided. We're very excited about moving forward with this as a way of revolutionizing the prognostication, treatment, and treatment for patients with liver cancer. Thank you very much for your attention.
Thank you.
So I'd like to thank Renu and Dean for this great webinar, and just for your reference, we'll dwell on this slide for just a moment. If you're interested in more information, please contact the licensing associate who is responsible for this innovation. That is Irit Gal, whose email address is shown here at the bottom. And this is docket number S14-354. So again, with that, we'll close the webinar. Thanks again, everybody, for attending.
Thank you.
Thank you.